Let me begin by expressing on my own behalf and that of my colleagues at MHRP our sincere condolences on the loss of your beloved King. I would also like to thank Diane for a fabulous lecture, and I hope I can maintain those standards for the morning. I only knew Joop and Jacqueline for a few years, but I'm sure they would have been an enthusiastic, double arm waving supporter for same-day treatment. He was a great advocate and a great researcher, and his impact will be felt for a very, very long time. I would like to thank the organizers sincerely for the opportunity to represent the HIV vaccine development field. I had a slightly different original title, HIV Vaccines, Are They Dead? Which we somewhat optimistically changed, and I think there's great reason for optimism, as you'll hear, and I hope appreciate in the following presentation. Is it a new beginning or the beginning of the end? So first, let's consider where vaccines stand in the growing portfolio of HIV prevention tools that we have. We have circumcision, of course we've had condoms and behavioral interventions, but importantly, pre-exposure prophylaxis, and perhaps with a resounding impact, the advent of universal treatment for everybody who's HIV infected on their own medical health care behalf will translate into lower incidence rates in the coming years. And so the question might be asked, and certainly is asked in some quarters, do we really need a vaccine? Does it merit the considerable investment in time and money to achieve an HIV vaccine? Now, keeping in mind that vaccines are the most cost-effective preventative medical measure that we have for most diseases and have a lasting impact on global health and throughout developing the developed world, the intuitive answer might be indeed, of course it's going to be beneficial. And in fact, models will show, as shown here in the solid lines, you see the current prevention package in green and what impact it would have in the coming years on HIV incidence. In orange is a increase but not full implementation of the existing prevention intervention modalities, and then in purple is if we fully implement all of the capacity that we would have for prevention exclusive of vaccines. And in the dotted lines you see what HIV vaccines would contribute to a reduction of HIV and in accelerating the end of this awful epidemic. So I think the answer is yes, HIV vaccines are worth pursuing, and indeed I think they could be a game changer as the years go by in terms of cost-effective means to reduce or eliminate the epidemic. So where are we? What have we done? Now, when asked by friends, particularly people outside of the field, how do you measure progress or what's around the corner? What's critical, of course, is the advent of efficacy trials where we really evaluate whether or not a vaccine works. And to my thinking, since the outset of the epidemic, what you see here depicted in this figure in blue is the time interval from the beginning to the end of efficacy trials and the vaccine components that have been tested or strategies that have been tested. And I think it's a shockingly and depressingly small list. What we don't know about how HIV vaccines, or in some cases any vaccines, really protect is rather shocking. And the only way we really learn is by conducting an efficacy trial and evaluating them very carefully. And we learn more from partial successes than we do from failures. And we have only thus far, in this relatively small number of studies, one partial success, but we'll spend some time discussing it since it's been leading the field and reinvigorated HIV vaccine prevention development. I would also add that on this list, 2 of these studies were conducted in Thailand, and one considers the size of Thailand, the size of the epidemic in Thailand. This is a disproportionate commitment by the Thai people and investment in prevention strategies, which I think is a shining example to other countries to make similar sorts of investments within their country, their scientific cadre and financial commitments to contribute to prevention development. And that's not just true for HIV vaccines, where they've definitely been leaders, but also in the development of other prevention technologies. So where are we today? Well, We have, broadly speaking, three strategies. As many of you know, most vaccines prevent disease, not infection, and they do that through antibody-mediated effector functions, and the most common is neutralizing antibody. Neutralizing antibody prevents the entry into a target cell or tissue of the pathogen. So from the beginning, there has been a desire, the holy grail, if you will, for HIV vaccines is a vaccine that induces neutralizing antibody that would be active against a large array or large diversity of the very variable HIV strains in circulation. But we've known from the beginning that most of our vaccine strategies, and this is true today, when administered to humans, do not produce antibody that neutralizes the kind of virus, the strain of virus that's transmitted from one person to another. They are only able to neutralize what we call tier 1 viruses, which are, for the most part, kind of special and unusual and adapted to the laboratory. On the other hand, and we'll go into this in a little bit more detail shortly, the Thai efficacy trial that was conducted here under the Ministry of Health, the Royal Thai Army, Mahidol University, and with collaboration with the United States Army, AFRAMS, and funding from NIAID, emphasized that you might be able to protect from HIV infection without inducing a neutralizing antibody response. A landmark study, and that is carried momentum into new efficacy testing that we'll discuss in more detail. Now, throughout the 90s and early 2000s, because we were not able to produce neutralizing antibodies with our vaccines, there was a lot of attention to the potential for a T-cell vaccine that would mediate CD8 killing of infected cells as a potential for either preventing infection or at least modulating disease in those who became infected. For the most part, those strategies have been abandoned. However, there is a single vaccine, not yet in humans, that uses a recombinant and attenuated form of CMV, and in non-human primate experiments is shown to protect from infection. Actually, all of the animals become infected, but 50% of them clear the infection. And it is beginning to creep towards human trials, and we will look forward to see how immunogenic and safe it is in those studies in the future. And then the last category is a new vaccine platform that consciously doesn't address the epidemic in a particular region, but rather has developed a strategy using mosaic inserts that are able to extend breadth of response across multiple subtypes globally. And this product also induces T-cell responses as well as non-neutralizing antibody responses, and we'll come to that later in the talk. So turning now to the Thai phase 3 efficacy trial, as you will recall, this study was conducted in Chonburi and Rayong provinces, and as is often the case in cohort studies as well as in intervention studies in HIV, you see greater incidence in the first year or 18 months of a given study, and then it attenuates. Those people who are at risk have become infected early in the study, and those that remain have a little bit less risk. And we saw that in RV144, and during that interval we saw protection from the vaccine. But as you can see in the upper left quadrant, the blue line, which represents the accumulation of cases in the placebo arm of the study, is parallel to that in the vaccine arm after about 1.5 years of study. In the right panel, what you have is a mathematical model of the efficacy of the study on a month-by-month basis, and the black line represents an estimate of efficacy, and above the midpoint line is positive. That is, the vaccine was working, and you can see that it works for a while and then goes away. Now, what we learned with a very simple set of analyses was that corresponded to a 15-fold reduction in peak antibody responses following vaccination, which is in the box to the right, perhaps a little bit difficult to see. And in fact, we published that the efficacy in this study was 60% at 12 months. It dropped to 44% at 18 months, but we followed people for 3.5 years after vaccination. And as cases accumulate, the net benefit of that interval of protection attenuated, and we end up with 31% protection overall. So our goal then is to further optimize the outcome of RV144, and we developed this model. So in the black line, what you see is the immune response, and the peaks correspond to the period shortly after vaccinations, and there's 4 vaccinations that are given in the RV144 approach. And we've arbitrarily drawn a gray line and a red line to represent low risk and high risk situations. And so you can envision a situation where your immunization strategy produces high quality responses, but they wane quickly, and so you're only able to protect high risk participants for a short period of time, low risk participants for a longer period of time, represented by the gray area end of the curve. But at the end of the day, your immune responses fall below the point where you're protecting anybody. So the question is, how do we improve the shape of this curve? Well, frankly, somewhat to our surprise, because the number of cases in RV144 were relatively small, we were nevertheless able to discern correlates of immunity. That is, we looked at people who received vaccine, and we compared those who became infected against similar participants who did not become infected, and we were able to tease out elements of the immune response that now serve as guideposts for further development of vaccines. And in particular, we identified antibody to the second variable loop of the envelope as a key correlate of a reduction of risk for acquisition. And I highlight this. There are many other correlates that have sort of been teased out of this experience, but this one also has evidence from a completely different direction to support its importance. And that is, if we looked at the viruses that infected people who received placebo versus those who got this vaccine, there's a difference, and that difference is in the V2 loop, suggesting this is indeed an important target for immune protection. And there was a sieve effect, if you will, a filter effect from those who developed antibodies to that particular motif. So based on that, we are going forward to try to improve the magnitude and frequency of that response to V2, amongst other characteristics, increase its breadth, because the problem you always confront with HIV is its genetic plasticity. It's quite diverse, even within a particular subtype. And then, of course, importantly, the durability of the response. So again, I would congratulate Thailand and all of our collaborators, the Thai Red Cross, the Ministry of Health, Mahidol University, and our core program here at AFRIMS for leading the way. We've done 3 studies in Thailand to carry forward on RV144, and it's provided critical information that's been influential in the design and development of the subsequent studies. I'm only going to refer to one today. This is RV306. Here, individuals receive the RV144 regimen, a vaccine at 0, 1, 3, and 6 months. And then they either receive no further boosting, or they receive boosting at 1 year, 15 months, or 18 months. I won't show you any data, but the outcome of this was to show satisfyingly, and perhaps not too surprisingly, that giving that later boost actually restored the blood levels of these antibodies of interest. Unfortunately, it didn't change their decay rate, but nevertheless, we can develop a mathematical model extending from the one I showed you previously, where at the time of in 144, about 60% of individuals had a response to V1, V2. Now with these boosts at 12, 15, and 18 months, we're able to see those levels or higher levels achieved with the boost. And so we can move a 44% efficacy observed at 18 months in RV144 to, we hope, 50% efficacy or greater at 24 months with an extended boost strategy. So the HIV Vaccine Trial Network that's run by Larry Corey and funded by the Division of AIDS at National Institute of Allergy and Infectious Disease has had a program of evaluating this strategy with a focus on addressing the terrible epidemic in Republic of South Africa or Southern Africa in general. Other studies that they've conducted in these locations have seen transmission rates of 6 to 9%. The first study they did is they took the same vaccine that we used here in Thailand, and they evaluated whether it would produce similar immune responses in Africa as it did amongst Thais. And the question they're trying to address here is, given the different conditions of endemic disease and genotype backgrounds and the host, would we still see the same frequency of response? And I'll come back to those responses, but they were very positive. In fact, they were the best responses that we've seen to that vaccine. VTN-100 was a pivotal study. It's still ongoing. It hasn't been finished, but the initial data is available to determine whether they would go forward into an efficacy trial or not. And the answer, which was revealed, I think, last May in 2016, was that they had achieved their benchmarks to go forward into an efficacy trial in South Africa called VTN-702. Those benchmarks were all developed around if the hypothesis of the V2 importance, V2 binding antibodies importance and efficacy is true, then what kind of response levels do we need to see in VTN-100 to test that hypothesis in efficacy testing? And they achieved those goals, and that study has started. So what are the differences between what we did in Thailand, which is shown in the bottom of this slide, versus what will go forward or is going forward now in South Africa? Is the same initial priming series, 0, 1, 3, and 6, with a boost at 12 months. But there are other differences here. It's a subtype C epidemic in South Africa, so they're not using the same vaccine. They're not using the vaccine that was designed to attack subtype B and subtype AE. So it's subtype C, 2 proteins from subtype C, and a canary pox that expresses subtype C. They also changed the adjuvant. We used alum in Thailand, and they are using MF59. So there are a few differences here, but the notion is to optimize responses against the V2 and therefore extend the sentinel evaluation observations here in Thailand. However, as time has gone on, we've begun to appreciate other aspects of that study in Thailand. Yes, we had 60% protection at 12 months. We need to extend that. 60% at 2 years is a product that would have benefit in certain clinical settings. But we then began to think about, well, what is the difference? And particularly looking at the response in the Africans when they used the vaccine that was tested in RV144. We began to appreciate that A244, the Thai strain that was in that vaccine, that envelope may have very important properties. It binds monoclonal antibodies that normally only bind conformationally intact trimers of the envelope. And those are the kinds of antibodies that are broadly neutralizing. We don't induce broadly neutralizing antibodies with this vaccine, but it does speak to the breadth of responses that we seem to induce. VTN-100 actually, while it met the criteria to go forward, was really not, it was a little bit inferior to RV144 in terms of the frequency and magnitude and breadth of response, and it was much inferior compared to the AIDS vax that was used here in Thailand when it was used in Africa. 097 was much better. So what has this led to? Well, it's reinvigorated the idea that we need to make a new GMP batch of that vaccine that was tested in Thailand. The breadth that's induced in 097 to subtype C variants of V2 is better than the vaccine they're using now. There's other cross-clade responses that are evident. And so a GMP batch of this material is under construction, and for our part at MHRP, with colleagues here at the Ministry of Health in Mahidol and in Germany, we're beginning to develop cohorts to assess what the incidence rate is going to be and the interest and support in communities for future vaccine trials, with a view to testing this new generation of products in a subtype B and in a subtype AE environment. So let me turn now to the AD26 plus minus MVA protein mosaic program. This program is led by Janssen Pharmaceuticals. They're enthusiastic supporters of this program. Their idea here is to develop a single vaccine platform that would be applicable throughout the global diversity of HIV, which is represented in the upper left panel by different colors representing different subtypes of HIV. The inserts are mathematically or computationally determined by evaluating a very large representation of available sequence data and defining insert sequences that have as much breadth as possible to cope with that genetic diversity. The one thing I'll say about this program, I didn't include any slides, but to the best of my knowledge, by far there is more non-human primate data supporting this platform, preventing SIV infection or SIV, HIV chimeric virus infection in non-human primates than other platforms that have gone forward, including the one we tested in RV144. So that's a reason for optimism. It's not 100% protection, but remember that animal model system is a fairly stringent system. Individuals get infected, individual monkeys get infected in an average of 3 or 4 exposures, and the risk per human exposure is substantially less. But also intriguingly, in these non-human primate studies, animals who break through and become infected often enjoy substantial suppression of viremia. And so this vaccine platform induces not only protective responses, but responses that help control the virus after infection. And so we are exploring them with colleagues at Thai Red Cross, HIV-NAT now as a therapy to achieve a durable remission of HIV. So here's a pictorial description of the vaccine strategy. AD26 is used as a prime at 0 and 3 months. Then there's boosting, and we have an ongoing pivotal study called Approach that looks at that boost as either AD26 again, MVA, modified vaccinia Ankara, a weakened version of a smallpox vaccine, and with and without a trimeric GP140 protein boost. The study is not yet complete. Analyses are ongoing, but the early results are encouraging, certainly for safety, and accordingly Janssen and its collaborators are planning a test of concept trial to be conducted in the Republic of South Africa in late 2017 or 2018 as a prelude to a program of global phase 3 trials that would include the United States, Europe, other parts of Africa, and we believe Thailand. So in my last section of the talk today, let me turn now to the advent of monoclonal antibodies. In the last 7 years or so, new technologies have allowed us to pull monoclonal antibodies out of individuals who have achieved broadly neutralizing antibody sera. Now, this is not common. It's only about 20% of HIV-infected people produce broadly neutralizing activity in their sera. It usually takes years. It typically is not associated with anything good for them. People who have those kinds of responses generally do not do better than people who do not have those responses. But it has been a transformative event in the field because it's identified targets for vaccination or for other programs that we might exploit in our vaccine development. But it also has begun to raise the prospect that we might have monoclonal antibodies that are highly potent, that can be used at reasonable doses, 1 to 5 milligrams per kilogram, that cover a very substantial swathe of the broad diversity of HIV. Now, it may require 2 or 3 monoclonals to truly be effective. We don't know, but early evaluations are ongoing. I'll come to that in a moment. But very importantly, there are modifications that can be made to these monoclonals that extend their half-life, so they might be an alternative in certain circumstances to antiretroviral treatment as a form of PrEP. They can be delivered subcutaneously to good effect. Whether they could ever be cost-effective compared to ART, though, is certainly an open question. But there might be circumstances where they're applicable, and we'll see how they develop. One of the first monoclonals, and one that was aggressively developed for use in humans, is VRC01. The Vaccine Research Center under John Muscola invented and developed this product. It's a monoclonal antibody to the CD4 binding site on the envelope of HIV. It has a very broad activity. It is very potent. It came from an HIV-infected individual who was a virologic controller for 15 years off of ART, so a little bit of an unusual case. since, as I said, most people who have these antibodies are unfortunately rather typical HIV-infected persons and don't benefit from the development of these antibodies. So here I'll show you just a little bit of data, VRC01 in a non-human primate challenge model, and there, as you can see, 6 of 6 animals that received 20 milligrams per kilogram were protected. Similarly with 5 milligrams per kilogram, partial protection with 0.3 milligrams per kilogram, proving the concept that this product can be delivered and prevent transmission of HIV, which is a chimeric virus with an HIV envelope and an SIV core. So based on this and other considerations, a collaboration between the HIV Prevention Trial Network and the HIV Vaccine Trial Network has gone forward called AMP, Antibody-Mediated Prevention. This is a proof of concept efficacy trial. The notion here is to prove that, in fact, neutralizing antibodies, as they do in non-human primates, will prevent infection, but more crucially, how much of that antibody is necessary to achieve that level of protection so that we can then begin to target that kind of response and calibrate vaccines to achieve those sorts of levels of responses. And also assess the importance of mucosal antibody versus systemic antibody and determine whether there's a realistic future for broadly neutralizing antibodies as a form of PrEP. This product will not go forward for licensure for that purpose. There are more potent monoclonal antibodies in development, and those also have the changes to the Fc receptor, which are associated with much longer half-lives, and they may go forward into studies for pre-exposure prophylaxis as well, or prevention as well. It's really 2 studies. One study is occurring in the Americas amongst MSM and transgender females, and the other is in high-risk women in sub-Saharan Africa, slightly different sizes based on difference in transmission rates, and overall 3,900 people will participate with a control for each of those 2 regions. So, in closing, we have several strategies that are entering efficacy testing now, and I hope to see that pace of efficacy testing of novel approaches to optimize these different intervention approaches accelerate. For non-neutralizing antibody, we've built on the work that was started here in Thailand with RV144 and is now in an efficacy trial, VTN702 in Southern Africa, and I hope in the not too distant future will be followed up in Asia and elsewhere. The combined T-cell vaccine under Janssen sponsorship is in a pivotal study, and we hope to see a proof of concept efficacy trial late in 2017. And now we have an efficacy trial for passive delivery of broadly neutralizing monoclonals. So great progress on many fronts. So let me close now by thanking the volunteers, those in Thailand, the United States, Africa, and globally. It is not a trivial commitment on their part. They're healthy people that have come into these studies and take an experimental vaccine that has some small risk of making them worse, and we cannot underscore the importance of their personal contribution enough. I'd like to thank the funders, the NIH, the U.S. Army, the Bill and Melinda Gates Foundation, the European Commission, and of course many of the host nations contribute vitally to this, and the institutions that are listed here. Thank you very much for your attention.